Okay, we're back at it again. I'll try to do this flipped classroom a little bit more. We're just struggling with time here with the new schedule. So this is on chapter two. I'm sorry, chapter 5, part 2, lipids. Friday I talked about carbs. You've seen that video. We discussed this in class for period G today. We'll probably get back to this with period E as well too today. So fats are hydrophobic molecules, meaning that they're hydro meaning water, phobic meaning fear. These are things that are non-polar, do not mix with water. And we'll talk about some of the basic principles behind lipids. They come in a few different varieties. There's things called steroids, cholesterols, and hormones that are one type or one classification of lipids, whereas glycerides or glycerols and phospholipids are a second. Store tons of energy, and the energy stored in these fats are actually in the hydrogen bonds, which you'll see as we go forward. So the fats can be basically there are two types of fats. Once again, they can be cholesterol steroid type fats, or they can be fatty acid glycerol based fats. Fatty acids. Here's a good example of one. This is a fatty acid. We have this long chain of carbons with hydrogens. That is the fatty portion, and the functional group, the acid group, the COOH, is the functional group. So the idea behind these is you have two basic structures. You have this glycerol molecule, and you have the fatty acid chains. One glycerol molecule can connect to up to 3 fatty acid chains. This would be a monoglyceride because there's only one chain. 2 chains would be a diglyceride. 3 chains would be a triglyceride. This particular one is called palmitic acid. As you can see, it's saturated. And the reason it's saturated is every carbon has as many bonds as it can between hydrogens. There's no double bonds between carbons, so it's completely saturated with hydrogens. What's going to happen, just as we looked at in sugars, is there's going to be a dehydration synthesis. All right, we'll drop an OH or drop a HO from either side. Either this OH or this OH can drop. It doesn't really make a difference. Water is removed, which makes it less polar, and a new bond is going to take place between the carbon and this oxygen, and it's going to be called an ester bond or ester linkage. So here's an example of a triglyceride. Once again, there are 3 fatty acid chains. Each chain is a long string of carbons and hydrogens. This is where the energy is actually stored, in these bonds between the carbons and hydrogens. We've talked about the carbon only makes nonpolar bonds because it shares equally with everybody, therefore not getting any pull or as we call the fat guy, skinny guy situation. So every carbon-hydrogen bond here is nonpolar. This whole section here would not dissolve in water. All right, great. The other thing with this triglyceride, once again, all 3 chains are saturated. Fatty acids, once again, they come in a saturated versus non-saturated form. These are saturated fatty acids. Double bonds between the unsaturated fats will actually create less chances for hydrogens to bond. So in this example here, we have our acid group at the end. Here's our C, the black triangle, OOH, COOH. The whites represent the hydrogens, and the blacks represent the carbons. Notice that when you have a double bond between carbons, you get this bend type of structure here. This bent shape actually causes these to be a liquid at room temperature as opposed to a solid. In general, saturated fats come from animals, and unsaturated fats come from plants. Here's another example of it. This is a really good example of what's called a phospholipid. So here's the glycerol molecule. You have 3 carbons in a row. All right. There, instead of having 3 fatty acid chains similar to this guy here, once again, this glycerol, there are only 2 fatty acid chains, and in this particular one, there's a phosphate attached to the very top. This is an example of a phospholipid, which are the main component of membranes. What I really want you to see here is that the fatty acid structures here, this one's straight, this one's bent, this one's straight, this one's bent. The double bond between 2 carbon molecules is what causes that bend. Okay, more hydrogens on here, more energy. So saturated fats do have more energy than unsaturated fats, but unsaturated fats, as you see the bend here, makes it take up more space, which we'll talk about in class about how that affects whether it's good or bad for you. Some other little pieces to keep in mind. So this is a phospholipid. The gray here at the very top is what we call the head. The head there. And then we have the tail, which is down here. All right, our tails. The tails tend to be fatty. The head tends to be polar. Okay, it gives cell membranes their particular um, characteristics, which in this case, you have your polar heads, non-polar tails. If you were to take these and toss them into water, just these phospholipids, they would organize themselves in this structure called a micelle. And the idea behind this is that there's a hydrophobic area inside, or water hating or water fearing area inside, and a hydrophilic area outside, which is where water would be attracted to. In cell membranes, we have two layers of these. So there's a double layer of phospholipids, which we'll get into more as we go. The big thing is to know that they look like this. They have a glycerol-based structure, have the two fatty acid chains, and one phosphate. So here's an example where the membranes would align themselves. Notice the fatty portions face in towards each other, and the polar portions face out. Number of reasons for this, but once again, this will be more of a class conversation. Steroids. Steroids have this basic shape. Kind of reminds me of a piece from the classic game Tetris. All right. So steroids have rings. There are 4 rings in all steroids. Steroids, hormones, cholesterol are all basically the same things. It's what function they have in the body dictates whether or not they get the name cholesterol, hormone, or steroid. So in this case, it's a steroid here, or cholesterol or hormone, that has the 4 basic rings. Each of these intersections here at the ring, which you're not seeing, would be carbon molecules. And then there are functional groups that are attached to these 4 basic rings. So once again, every hormone, every steroid, every cholesterol is going to have the 4 yellow rings here, and then have different functional groups that bond off of those rings. The functional groups are what gives it its function. Two major hormones that you probably know are probably estrogen, which is in females, and testosterone, which is in men. They're on-off. They tend to be on-off switches. So think about in puberty in young men and women, about the age 13, 14, hormones kick in. These hormones turn on things inside the body, and usually in men and women, it turns on growth at that age. So whether it be growth in height, body shape development, hair growth in men, facial hair, things like that. And once again, we'll discuss this a little bit more in class. I hope this helps. I know it's a lot to take in. We'll try these little quick videos like this as we go throughout the rest of the year. If you have any questions, please let me know.